water. In fact, a chemist will have chemicals around to remove the water that's produced there. And he certainly wouldn't have water in the reaction vessel. It would be the height of absurdity for a chemist who wants to make a protein to have water around. But do you think the ocean has any water around? If the ocean was a primordial soup, I'd say there's plenty of water around, which means the reaction's going to go this way. In fact, proteins and all the other things, they tend to break down in water. They don't form in water. The primordial soup, the primordial ocean, is the last place you expect to find this thing happening. It's just against basic chemical laws. Another thing we find here, supposing we have other chemicals reacting, instead of amino acid, you have something like this is ethylamine, and this reacts the same way, same thing happens, but you notice one thing about it, the chain is terminated there. And here's another example of that, acetic acid or vinegar. And once again, you've got a chain terminating there. And the point about, see, the Miller experiment produced about 5 times as many of these chain terminators as amino acids. So once again, you could never manage to extract the Miller experiment gunk and try to produce the proteins from it, because you've got all these other chemicals there which would just block the chain off. You could never get a protein chain growing in the stuff produced in the Miller experiment. So this, again, alone is enough to totally blow away the chemical evolution ideas. The other thing is the fact is the molecules of life, the sugars of the DNA and RNA, the proteins, they are all of one-handedness. You see, my left hand and right hand are mirror images of each other. And so are the different proteins. They are mirror images of each other. And the thing is, in living things, all the proteins are have amino acids which are left-handed. But the primordial soup could only produce an equal mixture of right and left. And to try and separate these things, you need to have something which is already handed. Organic chemists can do it because they've got plenty of things from nature which have one-handedness or the other to separate them. But in the primordial soup, you haven't got organic chemists around to do it. So once again, the primordial soup produces amino acids which are totally unsuitable. And here's the problem. You need to have all one-handedness to produce the proteins of life, and also all one-handedness of sugars to produce the DNA helix structure. And without the helix structure, it could not code for information or reproduce itself. And in fact, if you tried to have the wrong-handed form of the DNA building blocks, it actually blocks the chain off. A chain won't grow if you get the wrong-handed molecule there. Even a single impurity will actually block the chain from growing. Now, one thing we've got here is called the RNA world, and that's one of the ideas of modern origin of life chemistry, is that life began with RNA, which is a bit like DNA, but it's got a couple of differences, like the sugar's a bit different. The point is, how do you get the building blocks of RNA? You see, the sugars are very reactive. They need reactive formaldehyde to form. See, formaldehyde is used for preserving corpses because it reacts with proteins and cross-links them and makes them hard. It's a highly reactive chemical. You're not going to get formaldehyde accumulating on a primordial earth because it reacts with everything in sight. And the sugars themselves break down very quickly, and especially at high temperatures, because one idea is that life formed at these hydrothermal ocean vents. But the problem is, sugars break down at high temperatures really quickly. We have organisms, these extremophiles, thermophiles, they live in these extreme conditions. They have very efficient error-correcting machinery because the heat is breaking down a lot of their vital molecules, but they're repairing them as fast as they're being broken down. So you need these advanced repairing mechanisms before these high-temperature organisms can even live. And you need to have the sugars all right-handed, and sugars even react with amino acids, which is a bit of a problem. They're not supposed to do that, but in fact this is what the problem is, because that's how foods will often go brown. If you break the cells and you allow the sugars and the amino acids to react with each other, you are going to form browning reactions and not the right reactions you need for living things to form. You've got the bases here. So you've got two types of bases. Here's the pyrimides have one ring, purines have two rings. And the point is, one of these bases, cytosine, is incredibly unstable. There's no decent way of producing cytosine. And again, it's unstable at high temperatures. And look at the common reasoning we have here. And you find this in heaps of the evolutionary propaganda. You find traces of chemical A and B in the prebiotic simulations, and they're usually mutually incompatible. You can't have sugars produced in the same reaction that produces the DNA bases or which produces the amino acids. They are totally incompatible. But you find one experiment produces a trace of A, one has a trace of B. The next step is you get from a MaxiPure chemical company pure concentrated homochiral A and B from a chemical company, which has already done the purification for you, and then you react them to produce traces of C, and then you proclaim, well, there you go, we've produced C from primordial earth conditions. Well, in fact, there's been an enormous amount of intelligent design there to get the pure and concentrated forms of the chemicals to react them together. So it was never produced in these Miller experiments, and it never could be produced. And another big problem is, if you are trying to get the building blocks of DNA, you have to link the base of adenine here with the sugar, and it's very hard to do. But even when you have pure adenine and pure ribose and remove the water, because again, to link them together, they release water, so you've got to try and exclude water. Even under these favorable conditions, you can't get the sugar bonding with the one ring's amino acids. Okay, so even that's not possible. And you need phosphate to produce the backbone of DNA, and phosphate gets precipitated by things. Calcium would immediately react with phosphate and precipitate it out. And just to conclude here, to show you that these are only scratching the surface of the many reasons why life couldn't evolve from non-living chemicals. See, Yockey again, he said the paradigm of the primordial soup was worth consideration. Now the entire effort in the primeval soup paradigm is self-deception on the ideology of its champions. Then he says, in order to make progress in science, it's necessary to declare the decks to speak of failed paradigms. This must be done even if it leaves the decks entirely clear and no paradigms survive. And what he means is no naturalistic paradigms. But his belief in a primordial soup on the grounds that no other paradigm is acceptable is a fallacy of the false alternative. In science, it's a virtue to acknowledge ignorance. There's no reason that this should be different in the research of the origin of life. But the reason they don't want to get rid of primordial soup is because the only alternative